encroachment is occurring across the world. These blue dots on the map shows areas where shrub encroachment is intensifying. Right. Something wrong with this. Doesn't advance. Yeah. Okay. Okay. So on the other hand, there's a general increase that we are losing large herbivores at the global scale. And as you may know, a lot of these large herbivore communities are concentrated in the African continent, as shown by the coloration on this map. Conservation efforts can work to reduce those losses or even reverse those losses, which is really great. This map here shows variation in elephant populations in the African continent. The dark red colors and the blue colors indicate areas where elephant populations are increasing. So shrub encroachment is set to intensify specifically in Southern Africa, but also elephant populations are increasing there. And increasing shrub cover tend to slow or reduce grass cover. Specifically, if you think about the savanna, you think about open grass patches, but with increase in shrub cover, we tend to lose those open grass patches. On the other hand, increasing elephant density, for example, is linked with reduced number of big trees, which result to open savannas. So these savanna states, so on the one hand, due to shrub encroachment, you have a woody savanna. On the other hand, with increase in mega herbivores like elephants, you have an open savanna. And these two systems here have been shown to limit animal communities. I am using birds to try and understand impacts of shrub encroachment and large herbivores, specifically like birds, because they are easy to detect. They respond really rapidly to environmental change. They have a diverse set of traits that we can use to inform ecological models. Specifically, my dissertation was divided into 4 parts. So in the context of shrub cover, I had 3 main objectives. First, I wanted to look at long-term impacts of shrub cover. Secondly, I was interested in interspecies associations and to figure out if certain species were important in shaping those associations. And I wanted to zoom into a special nuclear species, which is called the sentinel. These species provide anti-predator benefits. So I wanted to zoom into this species to understand further how these associations are occurring in our savannas. And lastly, in the context of large herbivores, I was interested in direct and indirect effects of large herbivores. I did this in Kruger National Park in South Africa, and 4 parks in Eswatini. My study sites are located in a globally recognized biodiversity hotspot that is experiencing intensive land use. Kruger and Eswatini savannas are very similar in terms of climate, soil properties, vegetation composition. But what is different is that in Kruger, there's a thriving elephant population. Eswatini has not had elephants in decades. Instead, Eswatini is characterized by a thick, woody savanna. So this first part here concerns long-term impacts of shrub encroachment. So we know a little bit about how shrub cover is changing over time. There is some work trying to understand some of the drivers of shrub encroachment. But surprisingly, we lack studies on how these changes are impacting animal communities over time. So with that, we had these 3 questions here. we wanted to answer. At first, we wanted to figure out if shrub cover has changed in a 22-year period. Second, if those changes in shrub cover were impacting bird communities. Thirdly, we were interested in figuring out if certain groups of birds are experiencing those changes, those impacts more than others. So to do that, we collected satellite data in 1998, 2008, and 2020. We were specifically interested in quantifying the amount of grass, shrub, and tree cover in 48 plots that are located in 4 parks in Eswatini. So to quantify grass, shrub, tree cover in those plots, we used a random forest supervised classification algorithm in the Google Earth engine. And we also had access to bird point count data in 1998 and 2008. We went out in 2020 to collect more data. So because shrub cover and grass cover really exhibited this really strong negative correlation, so increase in shrub, reduce grass cover. So we excluded grass cover in our models. Instead, our model had the shrub cover and tree cover. So we used a multi-species occupancy model framework to investigate the effects of shrubs and trees, and we incorporated both linear and nonlinear responses. Because this talk is focused on parts of shrub cover, and also shrub cover had the strongest effects, that's what I'm going to show today. So what did we find? We found that in 22 years, shrub cover increased by 28% in our plots. As you can see in this figure here, the x-axis shows the 3 different periods. The y-axis amount of shrub cover. And as you can see, there's that general increase in shrub cover. But there's that variation in 2020 that we observed. We assessed variation of 64 species in total. And this figure here, down here, again, the y-axis shows species richness. It shows that at the plot scale, we tended to see that increase in species richness, again, with that huge variation in 2020. So trying to bring this together, this figure here, the x-axis shows the amount of shrub cover, the y-axis probability of occupancy, and the thin lines are our 64 species, so their responses to shrub cover. The black line, community response, and the gray ribbon is the credible interval region. So what we see here is that with increase in shrub cover, we tend to see a huge variation in species responses. But when you look at the community level, so focusing on that dark line there, we tend to notice that tapering and actually sort of going down a little bit with increase in shrub cover. So at the community level, we can say species occurrence start to decline with increase in in shrub cover. So we wanted to zoom into species that exhibited really strong peaks, and wanted to show that those species peaked at various amounts of shrub cover. And here, I just want to highlight 2 groups. The first group are migratory species. So these species overwinter in Southern Africa and breed somewhere in Europe, maybe parts of Asia. But for example, with increase in shrub cover, the barn swallow seem to tolerate really small amounts of shrub. And with the increase in shrub cover, we should expect this species to lose a lot of their overwintering range, specifically because they're already declining at the global scale. Their populations are declining. Another group are granivores. At the global scale, granivores are declining. In Southern Africa, also predictions indicate that this community is being replaced by woody adapted species. So again, with intensifying shrub cover, we should expect these groups to be further impacted. We wanted to zoom into specific groups. Specifically, we asked questions. We assessed effects of diet and nesting groups. And here, what we did is summarize the responses of these groups here. So for example, looking at diet groups, the x-axis shows the different groups, and the y-axis, the proportion of species that were responding in those groups. So the responses here, the light blue indicate positive responses. So the species are increasing in occurrence. Again, the curvilinear response, so that olive color there indicates those species that were peaking. So those exhibited strong peaks. That means they started declining with increasing shrub cover. Then blue indicate at the top indicate no response. So what I want to direct your attention to is diet groups such as things that eat seeds, fruits, and nectar. A lot of species there exhibit that peak response. So they might be impacted disproportionately by increasing shrub cover in the system. The same thing with nesting groups, things like, species that nest in cavities, for example, might be disproportionately affected by increasing shrub cover. So the take home from this part of my dissertation was that, of course, shrub encroachment is intensifying as predicted by other researchers, and it does seem to be driving a community-wide decline in species occurrence. And certain groups such as herbivores and cavity nesters are disproportionately affected. And with decline in things like herbivores and cavity nesters, we should expect impacts on ecological services and in general savanna resilience. So the second part looked at interspecies interactions, specifically in the context of mixed flock behavior, and also wanted to figure out if certain species are important in shaping those associations. So mixed flock studies have a long history in community ecology. So a mixed flock is nothing but two or more species moving and foraging together. So those interactions may have evolutionary population implications, and species may decide to participate in mixed flocks depending on benefits like predator defense or foraging efficiency. And these mechanisms may work hand in hand to influence species participation in mixed flocks. So mixed flock studies, usually, when we do mixed flock surveys, we are trying to characterize social network. A social network is nothing but species, in this case, species and their co-occurrences. So This toy figure here, the dark knots or these dots may indicate different species, and the lines or edges indicate their co-occurrences. So this social network here is characterized by three sub-communities. So there might be an underlying ecological phenomenon going on there, influencing this social structure. But in terms of our savanna bird communities, we expected that our social network would look like this, because we generally have granivores that forage on the ground, and we have insectivores that generally forage on woody vegetation. So the question was, how does our bird community, the question was, how is our social network structured? So we wanted to figure out if our social network looks like this. But the second question was trying to figure out if species that are associating in these social networks associate because they are facilitating each other, so they are similar and they are joining. But also, if these social networks, this social network is structured by species that are actually trying to avoid each other. The last part, again, we wanted to see if certain species are important in influencing those social dynamics. To do that, we went out to find mixed flocks in 2 parks in Eswatini. We used the gambit of the group approach. Again, this says 2 or more species moving and foraging together are associating. So what did we do? We went out looking for flocks. We counted individuals, we ID species. We also estimated foraging height in those flocks. After a few months of doing that, we wanted to figure out who usually dominate these mixed species flocks. And the idea there is that if you are found in mixed species flocks most of the time, you might be an important species. For example, you might be a nuclear species. So we did that and found that these 3 species here, the blue waxbill, dark-capped bulbul, and fork-tailed drongo, were very common in our flocking community. So we used these 3 as our potential nuclear species, and we wanted to test their influence in influencing species that usually participate in mixed flocks. The red-eyed dove did not participate in mixed flocks, so we used the dove as a control. So what was exciting here was that the blue waxbill is a granivore, and granivores are not usually associated with nuclear roles. So we're excited to test that. Also, the bulbul is a generalist. A generalist may or may not be a nuclear species. The drongo has been shown to be an important nuclear species in other systems. So we're excited to test the drongo. Specifically, the drongo is a nuclear species, but it is a special kind of nuclear species. It is a sentinel. Sentinels are important in predator defense, specifically provide that benefit for species that participate, that join the sentinel. So we use laid-back experiments to test the influence of these birds. To characterize our social network, we used 2 packages in R, and also did some social network analysis to investigate co-occurrences and to figure out if species to figure out how species are associating in those in that social network. We calculated association indices for these traits here: mass, diet, foraging height. Okay, and to understand the impact of the nuclear species, we use generalized linear models. So what did we find? In total, we characterized 92 flocks and 36 species participated in those flocks. On average, there was 6.6 species per flock. So this is our social network. So this shows more or less what we expected. So the yellow nodes are mostly granivores. There are some few insectivores, so they're mostly granivores, and the blue dots are insectivores that forage on tree tops. As you can see, there is that partitioning in our social network, but there's also a lot of interaction indicated by the red edges or the red lines. So there's a lot of interaction going on between these two communities. That was exciting to see. Zooming on to what is happening inside the social network. Again, this shows the 36 species that participated. And what I want to draw your attention to is the size, the difference in the size of the nodes. For example, this one with the red arrow. So the bigger the size of the node, the wider the community the species interacted with in the system. The smaller nodes, maybe they interacted with 2 or 3 species. Okay. On the other hand, some species really had strong bonds. So these edges here, the bigger the edge, the stronger the association between those species. So shown in blue arrows, these are species that forage on tree canopies, and they generally hang out together. They generally just stay together most of the time. And the black arrows indicate some granivores, also showing that some granivores were really found together most of the time. So that was really good to see. In terms of whether species that are similar participated or joined each other, or they avoided each other. So what we show here, for example, diet, foraging style being, are you chasing prey on treetops? Are you foraging in tree canopies? Are you foraging in cavities? Are you catching stuff in the air? So that's foraging style. And foraging height, where you found most of the time. So these 3 traits here indicate that similar species were joining each other. So we can say that our social network is structured by facilitation. So species are not competing for resources in our social network. So regarding the effect of our nuclear species, so our treatments elicited responses of 44 species in total, and 3 species responded to the dove, 15 to the bobo, 21 to wagsbill, and 26 to drongo. But what I want to show here on the x-axis, these are the different treatments. On the y-axis, number of species that came to the speaker, the trial. So again, this figure shows that our potential nuclear species elicited the strongest responses compared to the dove. But here, what we see also is that the drongo tended to attract more species compared to the bobo and the wagsbill. Also, the drongo elicited the highest number of responses in general. What was really cool was that these 3 nuclear species influenced really different species composition. Specifically, the drongo had a really completely different set of species that they influenced, with the bobo and the whitefish showing some overlap in their communities. So the drongo seem to have a distinct effect in this study. So the take home from this part here is that our social network is structured by two clusters, generally ground foragers and canopy foragers. And the association in the social network is driven by facilitation. So similar species are seeking to flock with others. Also that specific these different nuclear species influenced our community. For example, it was really cool to show that a carnivore is a really important nuclear species. I don't think that has been shown before, but also a generalist species seem to be an important nuclear species. Of course, the drongo was expected to have influence, and these three species here may be important in shaping bird associations in our savannas. So the next part, since the drongo had a really strong influence, we wanted to delve into the impacts of the drongo. And specifically, we asked two questions. Do the effects of drongo vary with shrub cover? Also, we wanted to test this notion that a nuclear species initiate mixed flocks. So we wanted to test that. In terms of variation in shrub cover, again, our prediction there was that with increase in shrub cover, the environment should generally drive heightened predation risk, because the environment is becoming more risky. More species may want to associate with a drongo that provide that anti-predator benefit. So we did that in, again, the two parks in Swatini, and specifically we searched for open and closed patches. This box plot here shows the shrub cover regimes in the two patches. So we selected plots based on the amount of cover that was there. So before we started any experimental treatment, we wanted to figure out the community surrounding our plots. Our plots were 20 meters radius. So we just got an overview of the birds that were around by doing a quick point count. And after that, to start a callback trial or a playback trial, we set two speakers. So in this toy figure here, indicated by A, so speaker A and speaker B, that were separated by 10-meter distance. And one observer hid about 10 meters from the speakers and had two devices connected via Bluetooth to the speakers. The idea there was, so we played maybe a drongo, a dove call, and the idea was to observe birds approaching speaker A. So birds that approach speaker A, maybe 10 meters radius, were considered to be interacting with the speaker. So we played a bird vocalization on speaker A for 10 minutes straight. And after that, we quickly switched and started playing the exact same tape on speaker B. Again, the idea was to see if species that came to speaker A were also moving towards speaker B. And if they did, there will be some evidence of following the drongo in this system. So we also, on speaker A, I also needed to mention that we estimated distances at which species interacted with the speaker. We analyzed that data using generalized linear models and generalized linear mixed models. What did we find? So in general, the background community was similar. So dove, drongo, playbacks were conducted on the same community. We counted 79 species in the background and 36 species responded to our treatments. This shows, so the figure, the box plot shows that indeed the drongo elicited the highest number of responses. So more species went to the drongo compared to dove, but there were no differences in the number of species that went to the drongo in open versus closed patches. Similarly, species composition, so species that came to the drongo did not vary between open and closed patches. However, there was some variation in the way species were interacting with the drongo. So it turns out when shrub cover increases, birds really wanted to associate closely with the drongo. So they were coming closer to the speaker as shrub cover increased. Did the drongo influence mixed flock behavior? So the x-axis shows responses to the first speaker. The y-axis shows responses to the second speaker. So there's this general very strong correlation. So species that went to speaker A also went to speaker B, indicating potential flocking in the system. So there were mixed flocks that were initiated by the drongo. And the take home from this part of my dissertation was that the drongo seemed to influence similar so there were similar communities that responded to the drongo in open and closed patches. Maybe because shrub encroachment is generally driving a widespread homogenized bird community, or maybe the open patches that are already there, that are still there, they are not big enough to host a distinct community. But with increase in vegetation, we did see that species wanted to associate closely with the drongo. That may have indicated that the environment is becoming more risky in terms of predation, and the drongo was providing that safety. And most importantly, we showed that the drongo does seem to form mixed species flocks in the system, which was really great to see. Okay, the last part, we're interested in direct and indirect effects of large herbivores. So we know quite a bit about the impact of large herbivores in general in our savannas. So specifically, we know that some herbivores, like mega herbivores, have marked influence on things like vegetation structure and animal communities. However, we really lack understanding of how these patterns come about. And one way to think about those potential mechanisms is through investigating direct and indirect effects of large herbivores. Specifically, we can think about indirect effects in terms of change in habitat structure or change in important resources like food. So this figure here shows the hypothesis, two hypotheses that we're testing. So we're saying large herbivores may indirectly influence bird diversity through vegetation structure. Also, we're saying large herbivores may influence bird diversity through changing food resources like arthropod communities. Specifically, here we're saying large herbivores have a direct impact on vegetation. Okay. At the same time, they have a direct effect on arthropod communities. But large herbivores may also impact arthropod communities through impacting vegetation structure. So we're testing this dynamics. To do that, we went to Kruger National Park, and there we are working in the browse plots. The browse project is a collaborative effort of these different institutions and organizations. So it has, so starting from the bottom, it has 6 plots that exclude mega herbivores like elephants. It has 3 plots that exclude all large herbivores. So from this, from a hare to the largest, the tallest herbivore out there. And there are 6 plots that are set aside as control. So what do? We are measuring vegetation in those plots, specifically grass, shrub, and tree cover. We are also counting birds. We are also collecting insects. We collect insects using sweep nests. So we use sweep net to collect insects on the grass layer. We also collect insects with the feeding sheet. So things that are found on woody vegetation, we use the sheet to collect them. And we take insects to a lab in Kruger National Park, and we count individuals under a microscope. We analyze that data using generalized linear models to understand the general patterns, and to understand the indirect and indirect effects of large herbivores, we use a structural equation model framework. So what did we find? So in total, we found 14 orders of insects dominated by these 4 orders here. So there was a lot of things like wasps, followed by grasshoppers, followed by spiders, and then beetles. We encountered 54 bird species. So generally, looking at the figure on the left, the x-axis shows the different treatments, and the y-axis patterns of abundance of arthropods. This figure here shows that when we remove a large herbivores from the system, we tend to see a decline in arthropod abundance. But if you look closely, it didn't matter whether we removed mega herbivores or all large herbivores. So that may indicate that mega herbivores might be an important factor here. In terms of variation in bird species richness, we did not see any variation between these treatments. In terms of vegetation structure, again, from the left, shows a variation in grass cover. The middle is shrub cover. So if you look closely here, grass cover, so variation in grass biomass, you see that when we remove large herbivores from the system, we tend to see that climb in grass cover. Again, the same thing happens with shrub cover. But if you look closely again, there's no difference whether we remove mega herbivores such as elephants or we remove all large herbivores. So again, things like elephants may have marked influence on these two vegetation components. In terms of changes in canopy cover, we tended to see an increase when all large herbivores were not present in the system. In terms of those direct and indirect effects that we're interested in, so again, these are our three different treatments. First, we looked at variation in canopy cover. So before I start, I should mention that this figure here, the black arrows indicate positive associations. The red arrows indicate negative associations, okay? And yeah, we did not show associations that were not significant. So all associations here are significant. So here we see that all these treatments were positively associated with increase in canopy cover. But again, there's that slight increase when we exclude all large herbivores. In terms of variation in shrub cover, there's that, so if we look at the partial exclusion there, we see a slight increase in shrub cover in those plots. So how did this then affect bird communities? We noticed that only a variation in canopy cover impacted species richness in the system. So large herbivores influencing canopy cover, influencing vegetation, influences species diversity by just changing shrub cover, canopy cover. On the other hand, when we investigated variation in arthropod abundance, again, we noticed that increasing canopy decreases insect abundance in the system, whereas this shrub cover seemed to be positively associated with the increase in arthropod. So remember that we said large herbivores may impact arthropod directly or indirectly through vegetation. And now we see this direct effect of large herbivores on arthropods. So in general, arthropod abundances did not vary in these three treatments. So, and how did that play out to influence bird diversity? Again, we see this strong association between variation in arthropod and species diversity. We were surprised to see that large herbivores may be directly impacting species diversity in the system. So, in conclusion, we showed that the herbivore community impact bird diversity indirectly via alteration of canopy cover and indirectly via impacts on arthropod populations. That was interesting that arthropod populations were associated with shrubs, but we think what was really changing is other things like forbs and other flowering plants that influenced this increase. But surprisingly, we also noticed this direct impact of large herbivores. Maybe the system has communities that really benefit from having large herbivores around. For example, shrikes that forage by following beta species, so species that are eating the vegetation, flushing arthropods. So things like that may benefit by following large herbivores. So I just wanted to generally wrap this up by saying most of our protected areas, especially in the African continent, are really existing in a sea of intensive land use. And we really need to think carefully about how we can facilitate the conservation in those parks, because we cannot afford to lose species. If a species can persist in our protected areas, it cannot persist anywhere. So if I was given 1 minute to wrap things up, I would say savannas that are experiencing shrub encroachment really need to be thinned. So we really need to have management practices that prioritize thinning of shrubs to enhance biodiversity. On the other hand, there's some research that is done, around the impact of elephants, and that we can somehow plant big trees and protect them until they're big enough to be less impacted by herbivores like elephants. So I would say we really need to plant big trees, because big trees seem to promote diversity in this region. Also, we need to promote long-term experiments, because this study, for example, was done in like 2 years, but this is a savanna. Also, context matters. So what if, for example, the communities have changed in such a way that it will be hard to perceive any strong changes in like 2 years? Maybe we need to have experiments that run for several years to be able to understand some of these impacts and patterns. It is also exciting to note that there are some talks about incorporating species interactions in ecological models, and that is really important, because traditionally species are just regarded as individuals, but generally everything is connected, species are interacting, and we really need to incorporate those interactions in our ecological models to understand changes in biodiversity more effectively. With that, I want to thank a lot of people. First, I want to thank my committee. I thank my lab mates, the Fletcher Lab. They're very critical in my success. I thank these individuals here, Dr. Lawrence Kruger, Dr. Wisdom Samini. I also thank a lot of volunteers in the Browse experiment. I thank students from Women in Science program. I want to thank Denile, who suffered through the process of counting and characterizing insects with me in the field. And I thank SNRE. Without the support, I wouldn't really have finished this research. So I thank the SNRE staff very much. I thank people working for these organizations and institutions. Thank you very much, and I will take any questions that you may have.
Any questions?
So you mentioned that certain bird species were more impacted by shrub encroachment than others, like the cavity nesters and the nectar feeders. I know that you said that one thing that would be good to do would be to incorporate interspecific interactions into modeling, but in the meantime, what do you think could be some of the impacts of loss of some of those different guilds of species?
Right. Yeah, so for example, if we lose things like cavity nesters, by the way, and cavity nesting birds are declining at the global scale, so there's a lot of work going on there. So not only do, for example, those birds make cavities for nesting, for example, but those cavities are used by other species for nesting, they're used by other species foraging, and so just using that logic, you can start to understand the ecological implications that it may have if you lose species that are really important in creating habitats for other species. You can start to lose things like we have like southern black flycatcher. So the southern black teat in our system, we are losing those because they nest in cavities, but they can't make their own cavities. Things like woodpeckers need to make this. So just making an example here. Yeah.
Hi, Muzi. Congratulations on your work and thanks for your presentation. I'm curious to know if, because you mentioned that the mega herbivores could be influencing the arthropods, and do you think there's also an influence in potential predators for the birds? Like the mega or the herbivores would be influencing predators of the birds?
So if there's something going on between mega herbivores and predators like raptors, for example. Oh, that's a good question. Yeah, I don't know. Yeah, I don't know, but I would think so. For example, if you look at those areas that are cleared of big trees, they have a lot of raptors. Just raptor activity there is really high compared to certain savannas. So definitely there is some interaction going on there. So by just changing the habitat in general, does change the community of raptors in the system. But yeah, if those changes in raptors also influence communities of passerines, that's the question that I may need to answer. I may need to provide an answer to. Yeah, but yeah, just changing the habitat does change raptor communities.
Yeah, one more question. Anybody online from Zoomland?
Oh, I think there's chats.